DNA sequencing is just like polymerase chain reaction. It's for DNA replicating. It's used in genetic diseases, to mutations, to pathogens for outbreaks, and genome mapping for many other things too. And first, you're going to isolate the DNA. So you start off with a single strand of DNA, and you have your fragment of interest, but you have your primers. And there's your primers. Now, what you're going to need is 4 test tubes. In each of the test tubes, you're going to need DNA, DNA polymerase, okay, and then you're going to need lots of DATP. So I'm going to draw lots of A's in each tube, lots of A's, lots of A's, lots of DCTP, so lots of C's, just everywhere, lots of DGTP, so lots of G's, lots of G's in the test tubes, and lastly, lots of DTTP. So lots of T's. Okay? And you're also going to need the didioxy, which is going to be, I'm just going to do them all black, so they're going to be DDATP. These are all the didioxy. So it's DDATP, so we're going to do weird A's in each one. You're going to need DDCTP, so just do a weird C in each one. DDGTP, let's put a G, G, and DDTTP, so a T. Okay? So now you're going to have your single strand of DNA with 1 primer. In this primer, you're going to have lots of other primers that are going to attach to this primer that is complementary, which is on a sequence of DNA. Let's say it's CTGA. It goes on to TTC G, C, T, A. So say it's supposed to stop at a didioxy A randomly. So this is your didioxy A, and eventually every A that is in the sequence, I'm going to change this G to A. So eventually every A that is in this sequence, or eventually every A that is in this sequence of DNA is going to turn into a didioxy A. So now the primer lays down, and now we're going to load this into a gel electrophoresis. Okay, so here's our gel electrophoresis. In this one, we're going to have A. The next one, we're going to have C. The next one, we're going to have G. And lastly, T. Okay, so see our gel electrophoresis is filled. And this is how it's going to look. For A, let's go to C. Let's go to G. And T. Okay, so how you read a gel electrophoresis, you always start from the bottom. So you start from these bottom ones, and you're going to read it up. So the very first in the gel electrophoresis is a C, and then an A, and then a G, and then a G, and then a T, and so on. So after you read it up, that's going to give you the DNA sequence to determine what you were trying to determine. And that is DNA sequencing.